Today we are going to learn the first chapter, Living World and Classification of Microbes. So let's start. Biodiversity and need of classification. So what is biodiversity? Biodiversity is the differences that exist between all the living organisms that are present on Earth and the need of classification. That is why we need to divide them into different groups based on their different characteristics. Last year we learned that all the living organisms on Earth have adapted, that is, they have changed their way of living according to geographic regions, food ingestion and defense, etc. So geographic regions is the different places. If they change the place that they are living in, how their body changes in order so that they can survive that different environment. Food ingestion, if they have changed their diet, then how their body changes so that they can digest the different food they eat. And defense, that is how they can protect themselves from different animals that want to harm them or use them as food. While adapting, many differences are observed in the organisms of same species too. So according to the 2011 census is when they count the different types of animals and birds and plants that are present in the different parts of the world. Around 87 million species of living organisms are found on the earth, including land and sea. So the different types of animals and birds and plants that are present either on the land or in sea are around 87 million different types. To study such a vast number, that is such a big number, because 87 million is a very big number, it was essential to divide them into groups. So because it was very difficult to study all these different type of 87 million types of animals and birds and plants, so therefore it was very important to divide them into groups. So groups and subgroups were created considering the similarities and differences among the living organisms. Similarities like whatever the same things were there in different types of organisms, and the differences that were there so that we can differentiate between different types of organisms. They used this to differentiate between the groups and subgroups of all the living organisms. This process of dividing living organisms into groups and subgroups is called biological classification. In history, Carl Linnaeus in 1735 divided the living world into two kingdoms, Vegetabilia and Animalia. So Carl Linnaeus was the first person who decided to divide it into two kingdoms, Vegetabilia and Animalia. So all the plants belong to Vegetabilia and all the animals belong to Animalia. Then Haeckel in 1866 considered three kingdoms, Protista, plants and animals. So later they found out that there was a different species called Protista and they didn't have anything to classify it to. It couldn't belong into Vegetabilia or Animalia. That is why they decided to add a specific group for Protista. Therefore now there were three kingdoms, Protista, plants and animals. In 1925, Chatton created two groups, prokaryotes and eukaryotes. Now this was a different type of classification in which Chatton he divided the organisms based on their cells. If it was a single cellular organism, then it was a prokaryote, whereas multicellular organisms belonged to eukaryotes. In 1938, Copeland divided living organisms into four kingdoms, Monera, Protista, plants and animals. Now by 1938, another group was found that was Monera. So they decided to make a specific subgroup for it. Robert Harding Whittaker was an American ecologist. In 1969, he divided living organisms into 5 groups. So now Whittaker was the one who divided all the living organisms into 5 different groups. For this classification, Whittaker considered the following criteria. So how did he divide all the organisms into 5 groups? It divided it according to the complexity of cell structure. So whether they were prokaryotic or eukaryotic. Complexity of organisms, if they had a single cell, that is unicellular, or if they're more than one cell, that is multicellular. Mode of nutrition, that is how they acquired their nutrition. Plants are autotrophic, that is photosynthetic. Auto means self, means they made their food by themselves using photosynthesis. Fungi are saprophytic. They absorb all the nutrients from dead organisms. So they'll grow on a dead organisms and absorb whatever the nutrients are stored in the dead organisms. Animals are heterotrophic and ingestive. That is, they ingest food from outside their body. Also, lifestyle was used to classify all the different organisms into different types. Plants were called producers because they produce their own food. They use photosynthesis to make their own food and store it in their leaves, fruits, etc. so that they can use it later for themselves and also so other animals can use it for themselves. Animals were called consumers because they consume their food from other sources. Animals cannot make their own food. Either they will have their food by eating other plants or other animals. Therefore, they were called consumers. Fungi are called decomposers because fungi grow on dead organisms. When a plant and animal dies, fungi grows on top of it by decomposing them. Also, phylogenetic relationship, that is how every organism is related to another organism based on their cell structure and types of cells. For example, prokaryotic to eukaryotic. So prokaryotic have cell structure which is very disorganized. So when the cell's structure gets organized, it becomes a eukaryotic cell. And unicellular to multicellular. That is, if when one cell divides into many cells, it becomes from unicellular to multicellular. Living organisms were either prokaryotic or eukaryotic. Okay, so prokaryotic had unicellular organisms like Monera. Okay, but Protista, which was a unicellular organism, could not be classified into prokaryotes because other features of it were similar to eukaryotes. So, under eukaryotes, they made two divisions, unicellular and multicellular. So, under the unicellular division came Protista. And under the multicellular division were fungi, plantae, and animalia. So, how did they differentiate which living thing was fungi, which living thing was a plantae, and which living thing was an animalia? So, if they had a cell wall, but the organisms couldn't perform photosynthesis, then they were classified into fungi. If they had a cell wall, but they also could produce photosynthesis, then they were plantae. And if they did not have a cell wall, they were classified under animalia. So, now we'll study each of the different kingdoms separately. So, the first kingdom we'll do is Monera. Activity. Take a small drop of curd or buttermilk on a clean glass slide. Dilute it with a little water. Carefully keep a cover slip. Observe it under high power of compound microscope. What did you see? Moving small rod-like microbes are lactobacilli bacteria. So, this is a type of bacteria which is present in milk. It's called lactobacilli. Lacto means milk and bacilli is the shape of the bacteria, that is like a rod. All types of bacteria and blue-green algae are included in the kingdom Monera. So, who all are included in Monera are all the types of bacteria and blue-green algae. Characteristics of Monera. All the organisms are unicellular. They may be autotrophic or heterotrophic, that is, they could produce their food by themselves or they acquire their food from outside. These are prokaryotic cells without distinct nucleus or cell organelles. So they have just a cell without all the normal cell organelles that are present in other cells. The next kingdom that we are going to study about is Protista. These include Volvox, Paramecium, Euglena and Amoeba. Activity. Prepare a temporary mount of 1 drop of pond water on a glass slide. So, if you take a glass slide and just put 1 drop of pond water on it. Observe it under low power and high power of microscope. So, when you observe it, what you will see is some motile microbes with irregular shape. So, as you can see, these microbes are very irregular shape and they are motile, that is, they will keep on moving. These are called amoebae. Characteristics. Protista are single cell organisms with well-defined nucleus enclosed in a nuclear membrane. So, as you can see, they have just one cell. They are single-celled organisms with well-defined nucleus. In this picture, you can see inside here is a well-defined nucleus. That is, we can see it very clearly. And this nucleus is enclosed within a nuclear membrane. They have pseudopodia or hair-like cilia or whip-like flagella for locomotion. You can see here this long tail that they have. So, this tail is called cilia. It is whip-like flagella and this is used for locomotion. That is, it is used for the organism to move from one place to another. So this flagella will move, and because of it, this organism can move ahead. They are autotrophs. Auto was self, so that is why they produce their food by themselves. Examples of autotrophs are Euglena, volvox, containing chloroplast. So chloroplast is the one that has chlorophyll, and this is required for the organisms to produce their food using photosynthesis. They can also be heterotrophs. Heterotrophs are the one that acquire their food from other organisms. So these include amoeba and paramecium. So these two protista, they eat other organisms, and that's how they acquire their food.